Dear students, today I will discuss on a single-stranded DNA bacteriophage called ØX174. Bacteriophages are viruses, and viruses are very specific about their hosts. Like some viruses infect animal cells, some viruses infect plant cells, okay, and some viruses infect bacterial cells. And since they destroy the bacterial cells after growing, they are called bacteriophages. Now, bacteriophages were first discovered by Frederick Tuort in 1915, and separately it was discovered by Felix D. Hedel in 1917. Now, depending on the genome of the bacteriophage, bacteriophages are called DNA viruses or RNA viruses. The DNA again may be double-stranded in nature or single-stranded in nature. The RNA similarly may be double-stranded in nature or single-stranded in nature. Now, most of the bacteriophages are double-stranded DNA viruses. Only few are single-stranded DNA viruses. Two families are there, Microviridae and Inoviridae. Microviridae, from the name it is clear, they are very small viruses and they have icosahedral symmetry. The capsid is icosahedral. And these viruses are lytic viruses. That means they infect the host cell, they grow inside the host cell, then they come out of the host cell by lysing the host cell, destroying the host cell. That is why they follow a lytic mode of replication. Whereas filamentous bacteriophages are non-lytic in nature. That is, they grow inside the host cell and then come out of the host cell without lysing them. Okay, and they do it by forming a pore in the membrane of the bacterial cell. They form the pore using their own proteins. So, they follow a non-lytic replication of mode, non-lytic infection cycle. ØX174 and S13, these are the two examples of Microviridae or icosahedral lytic single-stranded DNA viruses, and M13FD-F1, they are the examples of filamentous non-lytic single-stranded DNA bacteriophages. The Greek word micros means small. Since these viruses are very small, they belong to the family Microviridae. And they first provided the evidence of overlapping genes in their genome, and they first provided evidence of economy of genetic coding. And also the rolling circle mechanism mode of replication was first studied in this Microviridae family. Walter Fiers and Robert Sinsheimer demonstrated first in 1962 the covalently closed circular nature of the ØX174 genome, and Frederick Sanger and his team first sequenced the ØX174 genome in 1977, and this is the first DNA based genome to be sequenced. Arthur Kornberg used ØX174 as a model to prove that the DNA synthesized by purified enzymes in a test tube have the same nature as the DNA of natural viruses. And in 2003, Craig Venter and his group reported that they have synthesized in vitro a ØX174 genome from synthetic oligonucleotides. ØX174 belongs to Baltimore group 2 because it has a single-stranded DNA genome. It belongs to the family Microviridae and genus is Microvirus. ØX174 virus looks like an icosahedron. Okay, that is why it has icosahedral symmetry. That means it has 5:3:2 fold rotational axis of symmetry. Now, this is the structure of ØX174. The capsid is composed of 60 molecules of major coat protein F, and at the vertex, each vertex is composed of 5 G proteins and 1 H protein, and 5 G protein, 1 H protein together form a spike-like structure. So, it has 12 spikes, and these spikes are responsible for binding to its host. A small protein J is also found inside the capsid. The J protein binds to the single-stranded DNA and helps in the packaging of the DNA inside the capsid. Look at the size of the Microviridae, then the diameter of the icosahedron or the icosahedral head is 25 to 27 nanometer. The genome of ØX174 is a single-stranded DNA molecule of 5386 nucleotides, and it codes for 11 proteins. So, the genome length is shorter compared to its coding capacity.
Ma'am, if the genome length is short, then how can it code for 11 proteins?
Because of the presence of overlapping genes in ØX174 genome. If we linearize the genome, then we can see that gene A contains an internal translation initiation site to encode gene A star. Gene A product is required for viral DNA replication, whereas gene A star is required to shut down the host cell DNA synthesis. Also, the B gene is encoded within A, but in a different reading frame. K gene is also present within A, but it extends into gene C and is translated in a different frame. Similarly, gene E, which is responsible for the host cell lysis, is completely within gene D, but in a different reading frame. The termination codon for D overlaps with the initiation codon of J. Thus, they have coded all the proteins in their genome in overlapping fashion. Now, ØX174 recognizes lipopolysaccharides present on Enterobacteriaceae, okay, like E. coli, Salmonella typhimurium are the hosts for ØX174. And the minor code protein H, which is present in the spike, okay, acts as the ligand for the receptor. This H protein spans the capsid. Okay, thus a part of the H protein is present inside the capsid, a part outside the capsid. The outside part of H is responsible for binding to the lipopolysaccharide present on the host cell, whereas the inner part is responsible for ejection of the genome into the host cell. And at least one H protein enters into the host cell with the viral DNA. It is required for to initiate the viral DNA replication. Now, replication of ØX174 genome occurs in 3 stages. The first stage includes synthesis of the negative strand complementary to the plus strand.
Ma'am, what is meant by positive and negative sense DNA?
The sense of mRNA we consider as positive. So, the parental DNA having same sense as the mRNA is designated as positive, and its complementary DNA is called the negative sense. So, the genome present inside the phage1, the genome of ØX174 has the sequence similar to the mRNA. Thus, it is called positive sense, and stage 1 involves the synthesis of its complementary strand, that is negative strand. Formation of a primer is very difficult for ØX174. Now, 6 E. coli proteins initially form a complex called pre-primosome complex. This pre-primosome complex is formed by pri A, pri B, pri C proteins of E. coli origin and DNA T, DNA B and DNA C proteins of E. coli origin. Now, this pre-primosome is formed on the ØX174 genome. Pri A binds to a single-stranded portion of the genome and it acquires an ATP molecule. Now, primase enzyme of E. coli binds to the pre-primosome complex and completes the primosome complex. Now, using the energy from ATP hydrolysis, this primosome complex will slide through the ØX174 genome, and when they will get a priming site, they will halt. Okay. Now, DNA B molecule will alter the structure of that region and primers will be formed against that region. Now, E. coli DNA polymerase 3,1 ligase, they will complete the replication. Okay. And after one round of replication, one replicative form composed of one positive strand and one negative strand will be formed. Now, second stage involves multiplication of this replicative form. What happens? From the negative strand, transcription starts and all the phage encoded proteins are transcribed. One of them is GPA. This protein nicks the positive strand between 4305 and 4306 bases, 6th bases. Okay. and then E. coli replication machinery continues replication from the 3' end. Okay, and this mechanism is called looped rolling circle mechanism. Now, when the newly synthesized DNA will reach to the nicked site, again GPA will cleave it and unit length ØX174 genome will be released. It will now serve as the template for synthesizing the complementary strand. That means one more RF will be formed. Thus, when 30 to 40 RF molecules are formed inside the host cell, now the replication mode will switch from RF to positive strand synthesis, because when the new phage particles will be produced, what is required? The positive strand genome, which will be used as the genome inside the phage head, and capsid proteins. So, this switchover occurs when completed phage particles appear in the cell. Clear? So, when at a time when about 30 to 40 replicative forms are present in the cell, enough capsid proteins are also present. They will form the capsid and they will capture the positive strands, okay, and they will complete the phage particles. Now, completely RF formation will be stopped and phage assembly will start. So, the third step is asymmetric replication of progeny single-stranded DNA plus strands. Now, assembly process is quite complicated. What happens? 5 F proteins, that is major coat proteins, will assemble with 5 G proteins, that is spike proteins, and they will form a unit called 12S unit in presence of gene product B. Now, 12 H proteins or minor coat proteins will join and they will form the spike formation, and D protein forms a frame on which this 12S unit and H proteins form the prohead. Now, packaging of the DNA will start. For packaging, 2 proteins are necessary. One is C, another one is J. J protein binds to the DNA and helps in the packaging inside the head, but the entry process of entry of the DNA is mediated by the protein C. So, J and C in combined action, they enter the DNA into the capsid and then they package the DNA in a proper way. Now, A protein cuts the DNA when the head is full or the unit length genome is present inside the head, and D protein is released from the prohead. Now, the mature phage is formed. So, this picture shows how procapsid is converted to a virion when the single stranded DNA is packaged inside the procapsid. Now, when about 500 phages have been formed inside the host cell, they will lyse the host cell. For lysis of the host cell, gene E is responsible. Gene E codes for an endolysin. Okay. Now, once the E protein is formed, it goes and binds to the inner membrane of the bacterium, and it inhibits the activity of another protein, E. coli protein called MRAY translocase. This MRAY translocase is responsible for synthesizing lipid I. Lipid I is a precursor lipid for peptidoglycan biosynthesis. So, if when MRAY translocase activity is stopped, peptidoglycan synthesis is hampered. So, the membrane will become fragile and the newly formed virions can come out very easily. So, into the endolysin, the gene E product, it is indirectly helping the phages to come out, and the new phages will be released. So, today we have studied the life cycle of ØX174. It is a lytic phage, and after growing inside the host cell, these phages, they lyse the host cell, and mechanism of rolling circle replication was first studied in these viruses, and how rolling circle replication produces the new viral genome.